We have Dr. Horner. Dr. Horner is a scientific director for the Center of Neuroregeneration, co-director of the Center for Regenerative and Restorative Neurosurgery, professor of neuroregeneration, and a full member of the Research Institute at Houston Methodist Neurologic Institute. Dr. Horner received a PhD in physiology from Ohio State University in 1995. He did his postdoctoral training with Dr. Fred H. Gage and became a staff scientist in the Optogenetics at the Salk Institute in 1998. In 2001, Dr. Horner joined the faculty of the Department of Neurosurgery at University of Washington in Seattle. He directed a laboratory at the UW Southlake Union campus and was a member of the Institute for Stem Cell and Regenerative Medicine. Dr. Horner's research focuses on the role of glial and neural progenitor cells in the regeneration of injured and aging nervous system. Welcome, Dr. Horner.
Thank you, Dr. Caldas. It's a pleasure to be here. It's exciting to follow Dr. Chu. So in the spirit of this session being the future of stroke repair, I'm going to focus on sort of a forward-looking taste of several different technologies that I think are really going to impact and build on what you've just heard that Dr. Chu presented in the clinical realm. So I guess first I have disclosures. I am co-owner of a company, and I will point out where that applies in the talk. So it's a small company that's interested in one of the technologies I'll discuss. So these are the goals for this lecture. I'm going to talk to you a little bit about how electrical activity, what role it really plays in development and in repair. And then we'll talk a little bit about the different types of tools that can be applied for neuromodulation or stimulation of the nervous system and how they might be applied in stroke. And then at the end, we'll get real philosophical with my long-term bets on some, I think, technologies that are really on the horizon that may be potentially really impactful. So stroke obviously is a devastating condition that disrupts the nervous system. It kills cells, but one of its primary modes of long-lasting damage is to actually sever or cut nerve fibers in the nervous system. So for example, in this figure, if we just focus here, you're looking at fiber tracts that come from the surface of the cortex, go through the entire brain, decussate in the brainstem, and extend down to the spinal cord. So this major fiber population is the motor population or motor system for the control, the volitional control of our limbs. My primary interests are in spinal cord injury, but I like to make the point it doesn't really matter where you cut this pathway. So even if you're motor-centric like myself, if you have an injury within the brain or if you have an injury extending down through the spinal cord, we still have a major problem, and that is how do we reassemble the connections or reconnect those two parts of the nervous system, the area above and the area below the lesion. Now we have a big tough challenge right in the nervous system, right? Our nervous system has 100 billion neurons. On average, we're all walking around with 100 billion neurons, and it's estimated we have 500 trillion synapses. Some of us maybe have more. And so the big challenge is how do we recreate that complexity with something as simple as electrical stimulation or just stimulating the nervous system. So I'm going to kind of whet your appetite with just a couple of different concepts. So these are just papers that show two, I think, surprising findings of electrical stimulation, and they've been around for a while. The first article really just exemplifies something that researchers have been trying to figure out for a really long time, looking at molecular mechanisms, genetic mechanisms, and that is, why does the human nervous system grow so slowly? Why did we take so long to learn how to walk, and our nervous system took so long to develop? And it begs the question, if you have a stroke, do we have to wait? Do we have to recapitulate that developmental period? Well, it turns out electrical activity is really one of the only things we know that make nerves grow faster. So if you stimulate a nerve, it has a tendency to grow faster. Now, not as fast as we'd like to, even in the clinical paradigm, but it can improve the rate at which nerves can grow. Now, on the complete other side of the spectrum, I want you to think about what you can do when you stimulate the nervous system. And one of the things that you can do by stimulating a particular area, and we'll focus on that a little bit later, is that you can modulate a patient's sensation of pain, and as well, you can even modify quite radically their immune system. So particularly their peripheral immune system and how they respond to an injury can be dramatically affected. So neuromodulation offers a sort of a panoply of opportunities to promote recovery. So let's start first of all where I started, and that is learning neuroscience. And one of the most important figures, I think, in neuroscience is Donald Hebb. So Donald Hebb developed something called, we all refer to now, called Hebeian Theory. And for me, it's the easiest way to understand why, or the potential for using a neuromodulation approach to repair the brain. And that is that it's a very simple idea. It's simply, in a simple stated fashion, it's that neurons that fire together wire together. So if you look at the diagram you see here, there's a neuron A, and neuron A is faced with two neurons, B and C. If you look right above the neuron B and next to A, you'll see these dash marks. And the dash marks are the code or firing pattern of those two cells. And because A and B speak the same language, in other words, they fire about the similar frequency, they make a connection, they hold hands. C is singing a different tune, and therefore it doesn't connect. And that's basically, the easiest way to encapsulate development is that when you're learning to walk, it's the neurons that are firing in synchrony that keep you upright, and they start to learn that that pattern works, and they start making these connections. So we really want to get that type of regrowth and that very specific connectivity to be recapitulated right in the stroke patient. So a couple of things that are known about just administering electrical activity to the nervous system. I mentioned a couple of the articles. Some of the things that I think this is more of a highlight of what we don't know than what we do know. Dr. Chu mentioned a little bit about the use of high frequency and lower frequency stimulation of the brain for either excitation or actually inhibiting pathways. We know that for modifying the sensation of pain, we have to use really high frequency stimulation at about 100 hertz or higher, and that's one way we can block a patient's sensation of pain if that's applied in the right area. If we want nerves to grow, those really high frequencies aren't as effective, so we have to go down to a lower frequency. 20 to 40 hertz is what has been mostly studied. That seems to make nerves grow faster, and it's better at promoting new connections. So it's kind of what we call the plasticity range. But you can see just from those values I show in the text there that I wish I could add more value to that. We don't really know a lot more about the different types of frequencies and their value. If you think about motor systems, which is what we focus on, your motor systems really follow a much slower frequency stimulation, down in the 2 hertz range up to 10 hertz range. Motor neurons, the cells that actually project to your muscles, they can't even follow a train much higher than that. So we rarely stimulate in that realm, therapeutically. So only to make the point that the whole code, sort of the language of the nervous system and what makes connections work for either sensory systems or trying to rewire the autonomic nervous system for modification of the immune system, etc. We really don't know all that code yet. So we have to keep that as a future direction, I think, for science. So what does work? There's a lot of really great examples. I'm going to be focusing mostly on things that we've done here at Methodist, and that is, I'm going to ignore a little bit, of the vast literature out there. But I will say that a lot of the things I'm going to show you now are replicated in many laboratories around the world. So this experiment that you see here is in rodents. It's done in a rat with a spinal cord injury. Again, we're disrupting that motor pathway to the cortical spinal tract. This is the tract that extends from your cortex to your spinal cord. And if we disrupt that, if we injure it in a way that would be similar to a motor vehicle accident, but also very similar to a stroke, most of the damage we see in this area after a sort of a contusive type of injury really is, there's a watershed effect in the spinal cord where there is a spinal stroke around the immediate impact lesion. So what we've done in this experiment we've actually done two things. I'm going to focus on one, but we've put electrodes either on the surface of the brain, so we isolate, we figure out where the motor neurons are that project down to the spinal cord, and we can stimulate those with something we call a neurochip. Or we've put penetrating electrodes in the spinal cord below the lesion, and we find the motor neurons that project out to the hand and the forelimb, and I'll show you what that looks like in a moment. And if you stimulate either directly in the spinal cord or in the brain, and you stimulate for about 4 weeks in a rat, what happens is that you can take those electrodes out. The animals have significant improvement in hand function, which persists. There is a question again of dosing and whether you can do more, and I think Dr. Caldas asked the similar questions in the last session. These experiments are difficult to do, just as clinical trials are difficult to do. So there's so much things that we could think about adding to this that we don't know. So, but what does it look like? This is the behavior that we're trying to restore. So you see the rat. The rat has to reach through a narrow gap and grab a pellet. After an injury, the animal basically loses their ability to volitionally control, use their mind to control the hand. After stimulation for about 4 weeks, I'll show you, I think I can, let's see here, wrong button. Yeah, so if you look just at this part of the graph, you see the red bars are the stimulated animals, and this is a very specific measure of the improvement in their hand function. So they have a significant restoration. They go from really the inability to grasp a pellet and to use the volitional control to orient their body and retrieve it to about a 50% improvement from their baseline. So it's a really significant effect, and we can do this. Now, this is a controlled laboratory condition, and we put penetrating wires in the spinal cord, which is a relatively invasive thing. Can we do this in people, and can we do it in people with stroke in a less invasive way? So this is work by Dr. Sienko, who is in the Center for Neuroregeneration. He's a human neurophysiologist and works with patients. And what he's been doing is using, really asking the question, if you can stimulate across the skin, so percutaneous stimulation of the spinal cord, can you put electrodes in the same areas or similar areas that we're putting electrodes directly in the spinal cord? Can you do that across the skin and get significant improvement? And the answer is yes, and I think you can see from the video on the right, what you're seeing is a patient that is undergoing some assisted walking, and you'll see the muscle recordings on the bottom graphs, the purple in particular. Up above the green, you see when the stimulator is turning on, and what you can see is that there's a tremendous increase in the EMG activity in the legs during percutaneous stimulation. So this is an acute effect that while the stimulator is on, but working with individual patients, Dr. Sienko has been able to see an improvement both in standing and walking. So this is not full-minute complete repair, but it is an improvement, and it's non-invasive. So again, it's easier to apply. So what can we do? So Dr. Sienko has been working already to see if this will work in stroke patients, and has been having some early success. And so what does the future look like? I think the future looks like the great promises are in combining some of these methodologies, with rehabilitation, not beyond what I just showed there with just assisted walking. We also have the opportunity to combine multiple sites of stimulation. Why not stimulate, as Dr. Hebb would have done, the cortex and the spinal cord and try to drive those specific connections? So those are the kinds of things we want to work toward, using robotic stimulation of the limb even and the muscles. So a lot of things in the future that can, I think, make this technique even have more success. So I'm going to switch and talk about one more study that's was built here at Houston Methodist. And for that, I want to talk to you about just a little bit about the vagus nerve. So this is from the four inventors are on the right there. It's myself, Dr. Tatiana Wolf, Dr. Eugene Golanoff, and Dr. Gavin Britz. And what this was an idea that the group had to ask whether or not you could take advantage of the nervous system's ability to modulate the immune or inflammatory system, and specifically focusing on something called the vagus nerve. So the vagus nerve is a major sensory nerve in your body. It receives information from all of the organs in your body, and it really gives the brain the sort of the heads up, everything's okay or things are not good. And in reciprocal, the vagus nerve, through in processing of the brain, can send signals out to the immune system, whether to amp it up or to dampen peripheral immunity. In the case of stroke, there's actually a really remarkable amount of animal data to show that if you could stimulate this nerve in the acute phases of stroke, you would prevent a lot of the really damaging peripheral immunity and peripheral inflammation that comes on during the early stages of stroke. So it's been really well documented in animals. So the question is, can you do this in humans? One of the problems in humans is that the primary technology for stimulating this nerve is in the neck. So here's the state of the art, which is to do a surgical approach in the neck. And as we heard today, we know that in the acute ICU, it's challenging to do something like that to a patient. So the technology that's being developed at Methodist is based on the idea is to fit into the standard of practice or standard of care, and that is to put, as you see over here, put a multi-electrode array on the endotracheal tube. And it turns out the vagus nerve runs right alongside of the trachea. So this technology, we hope, will be developed for a clinical trial here at Methodist in collaboration with Dr. Volpe. So this is the latest step in that progression, and I think it's really an important part of the research, which is to move from rodents now to larger animals. So why do I say that? One of the primary differences and one of the big criticisms of research not translating from rodents to humans is that humans have a tremendous amount of white matter. So we started talking about that, how important the white matter is and how much white matter damage is a really one of the major factors for the devastation of stroke. And you can see on this diagram, if you look, the little teeny tiny brain up on the top left is a rodent brain. But what I want you to see is that a pig brain here is stacked on top of the human brain and the primate brain. That the pig brain has a tremendous amount of white matter, very similar to that in humans. And what you see set up here is a model of stroke in the pig that Dr. Britz and others at Methodist are working to develop as a testing bed for the technology I just told you. So it will certainly be useful for that, and also for Dr. Dmitry Sayenko to begin to try new technologies where we think it'll really have an impact for scaling and modeling for a better controlled clinical trial. So I'm going to finish with the future. This is something called Optogenetics, and Optogenetics is it's a very old discovery. People discovered algae, as you see in the dishes you see there. If the top panel of dishes, this is a type of algae that's growing in the ocean. If you shine a light on one side of the dish, the algae will migrate from one side to the other. So it turns out that phenomenon is driven by a channel that's light sensitive in the algae, and it's essentially like the algae's eyeball. So this channel, some very exciting research has shown you can take that channel and you can engineer it into a neuron, and this is now even being done in the human brain, in the retina in particular. If you engineer that channel into a human neuron and cast light on it, particular wavelengths of light, you can get that neuron to fire. We have a very good control of light frequency, and so that is I think a promising new technique. So it kind of gets around the idea of having to use penetrating electrodes and may give us a sophisticated way to create very specific patterns of stimulation depending upon the circuits that are lost and mapped. And in fact, on the lower right, you see there, that's a reality. Those are goggles that are being produced by a laboratory in Israel. There are also multiple companies that are producing these goggles in anticipation. So one of the lags for putting this into the clinic is actually the technology for making the camera to shoot the light onto the retina when these channels are engineered into the retina. So this is near future technology. It'll be probably almost certainly first in the visual system, and then I think we will see it impact in stroke. So the last thing I'll tell you is this is the science fiction. This is something called Sonogenics. So it turns out that earthworms know where they are in the world, and part of that is because they have a couple of different sensors. One is they can sense magnetic north, very sensitive magnetic channels. They also have incredible mechanotransduction channels, meaning they can sense vibration. And basically for them, like an orchestra must be an amazing panoply of sensations, because they have collections of neurons that can sense these different frequencies of sound. So I think in the future we're going to see technologies like this, in addition to optogenetics. One can imagine engineering human neurons to express these, and then using different frequency ultrasound, which actually penetrates the body quite nicely as a way to control the nervous system. So I'll leave it there, and I thank you very much for the opportunity to take us in sort of a trip into the future. Thank you.
Well, that was